Imagine you have lived your entire life believing you know exactly who you are. Your family has been rooted in the same country for generations. You carry a quiet pride in that connection. To the land, to the history, to the people who built the world around you long before you arrived in it. Now imagine a scientist hands you a result from a laboratory. A genetic analysis. A biological truth extracted not from memory or tradition, but from ancient bone fragments buried in the soil for nearly 2,000 years. And that result tells you that almost everything the history books said about your ancestors was incomplete, or wrong, or both. That is not a thought experiment. That is exactly what happened, not to one person, but effectively to an entire nation, when researchers began systematically comparing the genetic signatures of modern British people against ancient DNA recovered from Roman-era skeletons excavated across Britain. They expected to confirm a familiar story, a tidy story, the one that fills museum placards and school textbooks and travel documentaries, the one about Roman soldiers marching across a Celtic island, building roads and baths, then retreating back to their empire and leaving the native population largely unchanged. What they found instead was so far outside that expectation that it stopped historians mid-sentence. The data did not simply add nuance to the old narrative. It dismantled core assumptions that scholars had held for generations. It revealed a version of ancient Britain that nobody had imagined. A version hidden for 2 millennia inside the molecular structure of teeth and femur bones lying forgotten in museum basements and unmarked graves beneath British city streets. And the central question it raised was one that nobody had seriously asked before. Who were the people of Roman Britain, really? And why did science have to tell us something history never could? For generations, the standard story of Roman Britain followed a reliable and comfortable arc. In 43 AD, Emperor Claudius launched a full-scale military invasion of the island the Romans called Britannia. Within a few decades, Rome had established military and administrative control across most of what is now England and Wales. They built extraordinary infrastructure, straight roads cutting through forests, grand bathhouses in cities, amphitheaters for public spectacle, and of course, Hadrian's Wall stretching across the northern frontier. They brought Mediterranean culture to the edge of the known world. And then, around 410 AD, as the Western Roman Empire buckled under a cascade of political and military catastrophes, they withdrew. The legions pulled back. The governors departed. The Roman administrative machinery collapsed almost overnight, and Britain was left to manage itself in a rapidly changing world. The conventional assumption that followed from all of this was straightforward. Rome was understood as a foreign occupying force, militarily dominant, culturally influential, but demographically thin on the ground. The idea was that the Roman presence in Britain consisted primarily of a ruling class of soldiers and administrators who governed from a distance. They lived separately from the native Celtic population. They built their cities and their villas, they taxed and they legislated, but they did not mix broadly with the locals. They were, in genetic terms, a superficial layer on top of a population that remained essentially Celtic, essentially native throughout the entire period of occupation. When Rome left, according to this model, Britain returned to its roots. The Celtic population reasserted itself. Then came new waves. The Anglo-Saxons, the Vikings, the Normans, each one adding fresh genetics on top of that enduring ancient foundation. It was a clean story, structured, teachable, and it had exactly one catastrophic problem. The bones said otherwise. To fully grasp why this discovery was so unexpected, you have to understand the technological revolution that made it possible in the first place. For the overwhelming majority of human history, the idea of reading genetic information from ancient remains was not just impractical, it was impossible. DNA is a fragile molecule. Over centuries and millennia, it breaks down. It absorbs groundwater. it becomes contaminated by bacteria. It fragments into pieces so small and so damaged that reconstruction seemed out of reach. That changed fundamentally in the 2010s, when advances in next-generation DNA sequencing transformed the field of ancient genomics almost overnight. Scientists developed laboratory techniques capable of extracting and reassembling fragmented genetic material from just a few milligrams of ancient bone or tooth enamel. What had been science fiction became standard methodology. Suddenly, the biological past of human populations was readable in ways that no previous generation of researchers could have imagined. Major research institutions moved quickly. The Reich Lab at Harvard University built one of the world's largest ancient DNA databases. The Francis Crick Institute in London launched ambitious projects focused specifically on ancient British genomes. Collaborative teams across Europe began pooling data, standardizing methods, and expanding the scale of analysis. Within a few years, researchers had sequenced ancient genomes from hundreds of archaeological sites, spanning thousands of years of human prehistory and recorded history. The method itself is elegant in its logic. By sequencing an ancient genome and comparing it statistically against databases of modern and ancient populations from around the world, scientists can determine with remarkable precision where an individual's ancestors came from. They can detect admixture, the biological blending of two previously separate populations. They can track migration routes, identify population replacements, and distinguish between groups that looked similar on the surface but carried fundamentally different ancestries beneath it. When this technology was turned systematically on Roman Britain, it produced results that arrived in waves, and each wave carried a new and deeper shock. The skeletal remains examined in these studies were recovered from Roman-era burial sites across Britain, dated primarily between 100 AD and 400 AD, the height of Roman occupation. Researchers extracted DNA from dozens of individuals and ran the comparisons. The question was precise: how genetically similar were these people to the native Iron Age British population that had inhabited the island before Rome arrived? And how closely did they match populations elsewhere in the sprawling Roman Empire? The answers were not what anyone was prepared for. A significant proportion of the individuals buried in Roman Britain showed genetic ancestry originating not from northwestern Europe, not from the Celtic and pre-Celtic populations that had inhabited the island for centuries, but from the southern and eastern reaches of the Roman Empire. Researchers found ancestry tracing back to the eastern Mediterranean basin, to North Africa, to the Levant and modern-day Syria, to Anatolia, the Balkans, and regions surrounding the Black Sea. One individual buried in Roman London carried ancestry with clear connections to sub-Saharan Africa. Others showed genetic profiles closely aligned with modern populations from Egypt, Turkey, and the broader Middle East. Some individuals had ancestry consistent with the Iberian Peninsula. Others matched populations from what is now Romania and Bulgaria. The burial site of a high-status Roman woman in York, known informally as the Ivory Bangle Lady, had already puzzled archaeologists for decades due to the African ivory among her grave goods. The DNA confirmed what those artifacts had quietly hinted at. This was not the minor, marginal genetic diversity that historians had assumed. The Roman period population of Britain was genuinely, measurably cosmopolitan in the fullest biological sense. People from across an empire that stretched from the Scottish border to the Saharan desert were not just passing through Britain. They were living there, raising families, dying, being buried, leaving their genetic signatures in the soil for scientists to read 2,000 years later. And when researchers then compared this ancient Roman-era DNA to the DNA of modern British people, an entirely new layer of the story revealed itself. One that was, in many ways, even more surprising than the first. Here is where the story takes its most thought-provoking and genuinely sobering turn. The extraordinary genetic diversity that characterized Roman Britain largely vanished from the British gene pool after the Roman period ended. When Roman authority collapsed in the early 5th century and the legions withdrew, the demographic landscape of Britain transformed rapidly and dramatically. The Anglo-Saxon migrations that followed, successive waves of Germanic peoples crossing from what is now northern Germany, Denmark, and the Netherlands, reshaped the genetic makeup of the population in ways that were sweeping and, in historical terms, relatively swift. Studies drawing on dozens of ancient genomes from the early medieval period found that between 50 and 75% of the ancestry of people in eastern and central England by the 6th and 7th centuries traced back to these incoming continental Germanic populations, not to the Iron Age Celtic inhabitants, and not to the cosmopolitan Roman-era community that had preceded them. The process was not a simple military conquest followed by cultural assimilation. It was a genuine, large-scale demographic transformation, which means the vivid genetic diversity of Roman Britain, the Eastern Mediterranean lineages, the North African heritage, the Levantine and Anatolian ancestry, was largely erased, not necessarily through organized violence, but through the gradual marginalization and demographic submergence that follows when a large incoming population fundamentally restructures the society around it. The people who had carried those extraordinary ancestries, who had been born in Britain but traced their roots to Egypt, Syria, and the Roman provinces of Africa, faded from genetic visibility. Their descendants, if they survived at all in the population, became indistinguishable within the broader genetic profile that replaced them. The irony of this is almost too cinematic to believe. For generations, the debate about Roman Britain centered on how deeply Rome changed the island. The more profound question, one that nobody thought to ask, was how much of Roman Britain's actual living population survived at all. The implications of these findings reach far beyond a technical debate among geneticists and archaeologists. The first and most immediate consequence is a complete reassessment of what Roman Britain looked like as a living society. The visual reconstructions in museums, the scale models of Londinium, the illustrated history books, they have consistently presented Roman Britain as a society populated predominantly by pale-skinned northwestern Europeans who happen to have adopted Roman habits. The genetic evidence says that portrait is profoundly incomplete. The city the Romans built on the banks of the Thames was home to people whose ancestry spanned 3 continents. That is not a modern political claim being projected backward onto history. It is what the biological evidence, extracted from the ground beneath British cities, records directly. The second implication is a serious challenge to the concept of a continuous, unbroken British genetic identity stretching from antiquity to the present. That concept has cultural and political weight in Britain, as it does in many nations. But the genetic record shows that the population of the British Isles has been substantially replaced or transformed multiple times over the past 4,000 years. The Bell Beaker migration around 2500 BC replaced a large portion of the earlier Neolithic population almost entirely. The Roman period introduced remarkable genetic diversity from across the ancient world. The Anglo-Saxon migrations restructured the gene pool dramatically. Viking settlements in northern and eastern Britain added further layers. The Norman conquest of 1066 introduced yet another. Modern British DNA is not a simple, unbroken inheritance from ancient Celtic warriors. It is a palimpsest, a document written, partially erased, and written over again by successive populations across millennia, each one leaving traces, each one transforming what came before. That is not a diminishment of British history. If anything, it makes it more extraordinary. The third and broadest implication is what this discovery says about the Roman Empire itself as a human project. Rome is typically discussed in terms of its military power, its legal innovations, its engineering achievements, its eventual collapse. But here, in the genetic record of a distant island province, we see something else. We see evidence that Rome created genuinely multicultural communities at the edges of the known world. We see people from the Levant and Egypt building lives in York and London. We see children born in Britain carrying ancestry from the heart of Africa and the shores of the Black Sea. We see the Roman Empire not just as a political structure, but as a mechanism for human movement and connection on a scale that the ancient world had never seen before. That story was completely invisible for 2,000 years. It took science, not scholarship, to finally tell it. Every significant discovery answers one question and ignites 10 more. This one follows that pattern with almost mathematical precision. If Roman Britain was as genetically diverse as the evidence suggests, what happened to those communities in real human terms when Roman authority collapsed? Did they attempt to assimilate into the post-Roman British population that struggled to maintain order in the chaos that followed? Did some migrate back toward the Mediterranean, following the retreating infrastructure of the empire? Did they survive in isolated pockets, their ancestry diluted over generations until it became statistically invisible? The current evidence suggests all three outcomes occurred in different proportions in different places. But the full picture remains genuinely incomplete, and researchers are still working to reconstruct it. There is also a deeper philosophical question lurking beneath the genetics. Na does not carry culture, identity, or self-perception. A person buried in Roman York with Eastern Mediterranean genetic ancestry may have spoken fluent Latin, worshipped Roman gods without reservation, followed Roman legal customs, and considered themselves fully and completely Roman in every meaningful sense of the word. Their genome tells one story. Their lived experience told another entirely. Genetics can tell us where populations came from biologically. It cannot tell us who those people believed themselves to be, what they valued, what they dreamed about, or how they understood their own place in the world. That limitation is important. It keeps the science honest. It prevents the misuse of genetic data to draw simple conclusions about ancient identities that were, in reality, far more fluid and complex than any single data point can capture. And yet, the bones do not lie in the way that texts can. The molecules locked inside ancient tooth enamel survived 2,000 years of decay and burial to deliver a message that no historian, no matter how brilliant or thorough, could have reconstructed from written sources and material artifacts alone. The dead reached across 20 centuries and corrected the record. That is not a small thing. Step back from Britain now. Step back from Rome. Because while the specifics of this story belong to one island and one empire, the lesson belongs to everyone. Every nation on Earth carries within its living population the biological fingerprints of every migration, every demographic upheaval, every trade connection and cultural collision that shaped it across thousands of years. Every population that considers itself ancient and rooted is, in reality, a layered product of movements and mixtures that its own histories either forgot, simplified, or never thought to record. The British case is remarkable because the evidence is so sharp, so recent in archaeological terms, and so dramatically at odds with the prevailing narrative. But the same fundamental principle applies everywhere on the planet. Ancient DNA science is doing something that no previous historical tool was capable of doing. It is reaching past the written record, which was always incomplete, always filtered through the perspectives and prejudices of whoever held the pen, and reading the actual biological memory of human populations. It is giving voices to people who left no writings, no monuments, no names that survived. People who lived full lives in Roman Britain, who raised children and built households and participated in a society that was genuinely diverse and genuinely connected to a wider world, and who were then forgotten so completely that their existence had to be rediscovered by machines analyzing fragments of their teeth. The discovery that Roman Britain was cosmopolitan and genetically rich is not shocking because it was improbable. Looking at the nature of the Roman Empire, it was almost inevitable. It is shocking because it was invisible for so long. The truth was always there, encoded in bone and buried in soil, waiting for the moment when science would develop the tools to read it. History, it turns out, is not written only in stone and parchment. It is written in our DNA. And the story written there is older, stranger, richer, and more human than anything we imagined. We are only just beginning to learn how to read it, and what we find, every time we look closely, is that the past was never as simple as we needed it to be. The bones knew. They were waiting for us to ask the right question.